Hello, welcome to Meselson-Stahl demola, and today's lesson is going to be about DNA, replication, and structure. Guys, this is going to be so pivotal. They love asking questions like this when it comes to exam. So we're going to cover about the different nitrogenous bases we have, phosphates, deoxyribose, compared to RNA, and then we're going to get into replication where we'll learn polymerase, ligase, helicase, the whole lot. Let's get it done. Okay, so just to introduce, we should all be happy with DNA. DNA, deoxyribonucleic acid. It contains the genetic information of an organism, yeah? That is kind of your overview of what DNA is, but we need to break it down. Now, we would have done some biological molecules in topic 1. We would have covered carbohydrates, proteins, lipids, yeah? This is just another one of them. So, DNA is what we'd call a polynucleotide, which means a polymer of nucleotides. And here's a mononucleotide in front of you now. So, you guys always need to be comfortable with drawing this diagram and also saying the different parts of a nucleotide. So we've got our phosphate group over to the left, that circle, yeah? You've got the pentagon is a deoxyribose sugar. Now, let's make another distinction, yeah? This very specifically is DNA, yeah? Very specifically DNA. If it was RNA, we'd take out the deoxy and we just have the D and it's the oxyribose sugar. Or you could just even say just a ribose sugar, yeah? If we're talking about RNA. But for now, we're talking about DNA, yeah? Then we've also got a nitrogenous base. So these nitrogenous bases, adenine, thymine, cytosine, guanine, you might see them as A, T, C, and G as well. But in terms of the structure of DNA, that is kind of all you need to know. Yeah? In the structure of a DNA nucleotide, that's something that is, I'd say, likely to be asked in exams. Now, in RNA, very important point, your thymine is replaced by uracil. So you know your thymine replaced by uracil when you come to RNA. Just remember that. If it's in a question, if you're asked about like transcription or something, just say it, yeah? Transcription is not in this video, but I'm just putting the point out there. Now, we also just need to show this. So, I wanted to show you, this is a DNA molecule right here. I put a red dot next to it. That's just a DNA molecule, yeah? You can see the two separate strands, one's in blue, one's in yellow, and we've got bonds between the strands, yeah? I just wanted to show you in the diagram and then also to show you on this where I've drawn the nucleotides out, yeah? So in green, hydrogen bonds, they are going to occur between nitrogenous bases, yeah? So they will join the strands together. And you can see green is joining the two strands together. And then in yellow here, or orange, we've got the phosphodiester bond, which happens between adjacent nucleotides. So if you can imagine, say that on this side is the yellow chain and on that side is the blue chain, yeah? Between nucleotides in the yellow chain, you're going to have a phosphodiester bond, which is going to join it together. So I hope that makes sense. Now, also just to mention, between adenine and thymine, or between adenine and uracil, you're going to have 2 hydrogen bonds. And with cytosine and guanine, it's going to be 3 hydrogen bonds. I don't necessarily think it's the most relevant thing to know. It's just good to add on. And then those are also your complementary base pairing. So A to T, C to G, or A is to U if we're talking about RNA. Finally, this doesn't come up in SNAB normally, but it's just good to know. Purines, pyrimidines, I use the acronym pure ascald, yeah? So purine and then A, adenine, gold, guanine. So your purine bases are adenine and guanine, meaning that your pyrimidine bases are going to be thymine and cytosine and uracil as well. I just thought that's good to mention, good to know. Okay, so all of that was groundwork to talk about what we really want to cover today, DNA replication. Very complex topic, but guys, just follow it along, memorize it, make sure that you know this whole process because it's very, very important and can even get you up to 5-6 marks in an exam. Yeah? Cool. So, DNA replication, literally, the process by which DNA copies itself. And it will normally do that for mitosis, for example, not covered in this video. That will be in later. And then we've got repairing DNA as well. It's something that we can do DNA replication for. So, let's break it down into parts. Number 1, DNA helicase breaks hydrogen bonds. So you can see here on this side, and just know it's going to come across the whole strand. So maybe DNA helicase starts there, but it's going to go across the whole strand, yeah? It breaks those hydrogen bonds. Remember, we said the hydrogen bonds are going to be between the bases on different strands, yeah? What you form, you can see here, you expose bases on the strand. So that strand, you see it's starting to unwind. Your DNA is starting to unwind. That's a good term to use. DNA is unwinding, yeah? So that's the first step. And that's done by which enzyme? Let's highlight it. DNA helicase. Cool. Let's keep moving. So now both strands are separate. Yeah, you can see in the black, both strands are separate. This is a very good term. complementary free-floating DNA nucleotides are going to line up with exposed bases on the template strands. So you can see here in the blue, the complementary free-floating DNA nucleotides, they're lining up with the complementary bases on the template strands or the original strands of DNA. Now, maybe a point to mention in an exam, I just mentioned some of the complementary base pairing. So A to T, C to G. Just mention it. Very good. Could potentially pick you up a mark as well. Now, if we look at number 3, DNA polymerase catalyzes the formation of phosphodiester bonds between adjacent DNA nucleotides. And really emphasis on this word, adjacent, first of all, and 2, the enzyme, DNA polymerase. That is causing the formation or catalyzing the formation of phosphodiester bonds. Just make sure that you are saying these terms because they're really, really important when it comes to answering. So you can see here in the green, I've shown you what the action of DNA polymerase is doing. You see the exposed bases, they're just joining it together so that we have a new strand of DNA. Cool. Now, DNA ligase catalyzes the joining of the new strands of DNA with the original strands. So you can see here, you've got a green strand with a black strand. So at the end of DNA replication, we've got a new strand and an original strand making up a new DNA molecule. And we're going to get 2 new identical DNA molecules. That is the process of DNA replication. Now, what we'll call this is semi-conservative replication. And that's going to lead on to our next slide. But semi-conservative replication is the method by which they do DNA replication because you get 1 new strand and 1 original strand. Let's go and see some other theories about DNA replication. So that's always in science. We adapt, we search, we test, and we make theories. And for DNA replication, there were 3 main theories that were kind of going along before this Meselson-Stahl experiment. And they were conservative, semi-conservative, and dispersive. So if you look here, in conservative replication, what was the whole rationale? It was just that you have a DNA molecule, it replicates, you get an original strand and a new strand. So original in black, new in blue. 1 original strand, 1 new strand. That was how DNA replication occurred according to the conservative method. Semi-conservative, or the one we've accepted, and we're going to show why we've accepted it. But in the semi-conservative, you can see the DNA molecule splits off into 2 new DNA molecules where you have a new strand in blue and an original strand in black. Happy with that. And the final method, a bit of a funky one, yeah? It's dispersive. So dispersive is that we've got the DNA molecule and it splits off into 2 new DNA molecules, but you can see that all of the new strands are kind of interweaved with each other. Now, I would say in an exam, they probably won't ask you about like how one of those methods would work if it's not semi-conservative, yeah? They don't really care too much because they've been proven wrong. And this is how we're going to prove them wrong. It comes down to 2 goats of the game, Meselson and Stahl. So what they did is they got E. coli. Really, it's a bacteria that has a very, very high replication rate. I think it does 1 generation every 20 minutes. So they got a nitrogen-15 growth medium. And if you do chemistry, you'll know that the most frequent occurring form of nitrogen is nitrogen-14. So we've used a different isotope or a heavier version of nitrogen. We'll call this heavy nitrogen. Yeah. You put E. coli in a medium containing that. And why else have we seen nitrogen? Well, you're going to need nitrogen to make those nitrogenous bases for your DNA. So if they've used nitrogen-15, you are going to be able to tell that. Hence, we're going to grow E. coli in a nitrogen-15 growth medium for many generations. And that means that all of our DNA is going to have that heavy nitrogen-15. Yeah. Now, does a machine and apparatus, I'd recommend you to search it up, but it's called a centrifugation chamber or centrifugation tube. And essentially what it is you have a tube and you literally spin it rapidly, yeah? And you form distinct bands based on the mass. Cool. So you centrifuge the DNA and you mark the position of the heavy one. So heavy should be nearer to the bottom, right? So you're going to see a band of material. It will be DNA, but you're going to see that near the bottom of the test tube. Now, what did they then do? They transferred into the E. coli into a nitrogen-14 growth medium. So you can see it was all using the nitrogen-15. Happy days. Now all this DNA has nitrogen-15. Then Meselson and Stahl said, let's shift it into a nitrogen-14 growth medium because it will then use that nitrogen-14 to make more DNA. And we can tell what's the difference between the original DNA and the new one. So that's the rationale. So they transferred it to the nitrogen-14 medium. Like we said, grows really rapidly. So each generation is only like 20 minutes. And they let it to replicate for another generation. Then they said, okay, let's extract DNA. Let's centrifuge again, spun it around, and they marked the position of the bands now. I'm going to show you the results in a second. And literally, repeat that, yeah? Put it back in the nitrogen 14, go it for another generation, centrifuge again, keep it going. Let's kind of see our results. And this is what they saw, yeah? And we'll explain why this conservative in a second. But in Gen 0, or the ones going in the nitrogen 15 growth medium only, you can see the band near the bottom of the test tube, yeah? In the first generation, what they saw, a bit bizarre. They saw that the band had moved up to around the middle of the test tube. So you can see this is when they started to use a bit of nitrogen 14 to make their DNA. Cool. In generation 2, even more bizarre, we saw 2 bands, but it was almost like they halved the thickness of the big band in Gen 1, and they split it between one that's still in the middle and one that goes to the top. And what we can see there is that in the middle, that's a hybrid of nitrogen 14 and 15, but at the top, that's just nitrogen 14 or light DNA. And that confirms our semi-conservative method, because if you think about it, each time it's going to use one original strand and one new strand. So if we're doing one original strand and one new strand, just bear with me for a moment. In Gen 0, it's going to be both of the original. I know my diagrams are really bad. Apologies for that. Then in Gen 1, the new DNA molecules made, as we've seen, they're going to look like this, right? With the new strands and the original strands. And I haven't used the same blue. Apologies, I'm just going to get it out. But then, again, they're going to use a new strand and an original strand. But in the context of generation 1, that means that you're going to have this going on, where we have original strands like that, and we're going to have this one. We only have 2. We only have 2 original strands of black, yeah? So again, I'm just going to quickly fix up. We only have 2 original strands of black. Those are all our original strands. So we're going to add on new strands, which we made using nitrogen 14, which is why we're going to have some that are purely nitrogen 14. I'm going to have some which are now going to have a hybrid. But as we continue going through generations, the amount of hybrid DNA is going to be less in comparison to the ones that's purely just light nitrogen. I hope that makes sense. And I've also just drawn here what you would expect to see if we didn't have semi-conservative replication. So in conservative, you will literally just have 2 bands, yeah? Because they're just using the DNA, they're just making new ones. So you literally just have 2 bands, where you've got nitrogen 15, nitrogen 14, yeah? And then in dispersive, all of it would end up coming to the middle, because it's always going to be a mix, yeah? So I hope that makes sense as to what you would expect to see in a Meselson-Stahl experiment. I think especially if you're on SNAB, make sure that you're very comfortable with this experiment. Okay, question time. So I hope you can see this quite clearly. We've got 2 multiple choice ones there, which shows the components that can be found in the DNA molecule. And then they've said that 70% of the bases are guanine, what percentage are thymine? And then the second question here, compare and contrast the structure of ATP and a DNA molecule. And they've just shown you an ATP molecule there. So have a go at these questions, and then we'll get back into it. Okay, so now here are the answers. Now, my go-to whenever I see a question like this is, let's do a process of elimination, yeah? So, in a DNA molecule specifically, well, glycosidic bonds, Stefan and carbohydrates, yeah? Let's cancel off A and B. Now let's look here. They've said that uracil and even ribose as well, those are both going to be RNA, not DNA. Hence, we can get our answer that C is going to be the DNA molecule. Now let's look here. Well, we've got to think about our complementary base pairing rules, right? We've got to think about the complementary base pairing rules. So, if you think 17% are guanine, then it must be that 17% is also with the complementary base, which is cytosine. So we've already got 17 plus 17, 34. 34% of them between cytosine and guanine. Now, 100 take the 34 means that we've got 66% of the DNA left. Cool. Let's think about this. We know that in a DNA molecule, there's going to be an equal amount of adenine and thymine because they bond to each other. Hence, of that 66%, we half it to get 33%, which is going to get us the answer for B. Now, this question, a bit more interesting. So they've given you the structure of an ATP molecule, yeah? And they said compare and contrast, and you can see the points that they put here. What I would just say is, guys, let's make this a bit easy for ourselves. Literally, draw out a DNA molecule right next to it. Doesn't even have to be a good drawing. But I've drawn out my DNA, now I can start to compare. Well, this one's just adenine. I can have adenine, I can have thymine, I can have cytosine, I can have guanine for this one. Ribose, well, I said this is deoxyribose. Yeah, this is 3 phosphates, this is 1 phosphate. So we've already got a bunch of differences. What may be a similar? Well, adenine could be a similarity. Also, pentose sugar. Ribose and deoxyribose are both pentose sugars. So we can see what they put in the mark scheme, but yeah. You see another similarity, let's say here, highlight it. Phosphate, pentose sugar, and a base. All of them had that. Yeah. And then I think these are stuff that we mentioned. Deoxyribose versus ribose. DNA has other bases. And then ATP is only adenine. And you can also see that there's one phosphate versus 3 phosphates. So those are the kind of things you have to do. I think drawing out a diagram is really helpful with a question like that. And let's finish off with the A-star tips. So won't keep it too long, but literally knowing how to draw out that DNA nucleotide is going to save you so much times. Literally just be able to do it. Hydrogen bonds, phosphodiester bonds. With biology, you're going to learn about a bunch of different bonds. So literally, you need to be able to pinpoint where's this bond? What does this bond form between? Make sure that you are very comfortable with knowing that. So no phosphodiester bond is between the deoxyribose and the phosphate. Yeah. Know that the hydrogen bond is between the nitrogenous bases. Also, guys, this is like the classic. The enzymes in DNA replication, they're a bit of a mind boggler. So make sure that you know where each one does. We know helicase breaks the strands, ligase joins the strands, polymerase phosphodiester bonds. Yeah, make sure that you are so happy with that. And even when we get into transcription, guys, you need to remember sometimes you're gonna have to say like RNA polymerase. Yeah, so make sure that you're happy with that. Stages of DNA replication, guys, please just try to have a logical chain when you're answering a question like that. Now, replication theories. I think when we get to here, we're kind of a bit more specific to SNAB, but they do come up in other examples as well. I just think it's more common to be asked in SNAB. And also, AQA and OCR hasn't been covered in this video, but Okazaki fragments, leading and lagging strands, and 5' and 3' ends. In SNAB, you don't really need to know much about this, which is why I skipped it for this lesson. But if you are in AQA or OCR, make sure you have a review of that. What not to do. I've already mentioned this, confusing the enzymes happens a lot. Be careful. Confusing DNA replication with transcription happens a lot. Guys, be careful. Ignoring Meselson-Stahl. For some people, they open the exam board and they're like, I've never seen this. I've never seen this in my life. So make sure you're not one of them. Yeah. And that is my tips for this lesson. So we're going to wrap it up there. A bit of a longer one, I believe, potentially. Let's see what happens in the editing. But guys, if you enjoyed the video, please like, subscribe. Drop a comment below if you need anything clarified or anything like that. We've got the TikTok and the Instagram both at A-star demola, and I'll catch you in the next one. Bye for now.